Hi, everyone. My name is Hyun Kim from SNJ Pharma. Let me tell you about SH1, the first safe clot buster. Every 30 seconds, there are new cases of stroke and heart attack, totaling 240 million worldwide, affected by this life-threatening disease. All these four diseases are number one death of cause. And the underlying common cause is thrombosis. Of course, thrombus can be dissolved by drug. It's a thrombolytic drug called as TPA. It's a $1 billion blockbuster, but only 3% of patients is able to receive it. Just because 97% are not just qualified for this TPA treatment. Why? TPA is just not safe. It causes more than 10 times of a bleeding risk compared to placebo. So actually treating with the TPA could convert ischemic stroke to hemorrhagic stroke. So there are these long list of exclusion criteria for TPA. And in addition to that, we also have inclusion criteria for TPA, which means patient to be arrived to the hospital within 3 hours since the onset of symptom. And also, even after passing through all these criteria, patient treatment with the TPA is not that effective. It's going to dissolve just 30% of the clot. So to provide a solution for this deadly disease, we provide SH1, which use totally separate independent pathway, a safe pathway, as we published in this publication. Our data clearly confirm that it dissolves clot. And also, it dissolves large clot, unlike TPA. And most importantly, this one is safe. As you can see here, our bleeding assay shows that bleeding time, bleeding volume, clotting time is almost like a placebo with our drug, unlike TPA, the current only drug for stroke. So our program this year received the NIH grant with the top score and outstanding review, such as it would broaden patient population. It would broaden patient population that qualifies for acute treatment. So we plan to move to the clinical trial stage within 3 years. After approval with stroke, the following will be used for off-label drug use. So to make that happen, we might need $20 million for the use of 3 years of preclinical and clinical development. And these are our teams of founders and collaborators. This is a summary. Thank you.
Can you talk about what are your what the target is? What is the drug targeting? The mechanism of action of the drug?
Patient group targeting.
What's the mechanism of action? Can you go into more of the mechanism of action of the drug?
Actually, yeah, TPA works through by activating a plasmin, which is a non-specific protease, but our SH1 is plasmin independent, which means SH1 doesn't generate a plasmin, which is non-specific, but this SH1 is itself a serine protease, which is specifically recognized thrombus, and then digests this fibrin network. So that's the mechanism.
Some questions for you. If you go back to the slide where you're about the ask. No. Forward. where you're asking for the money, you're saying it's $20 million?
Oh,
So you've raised one to date. You're seeking a 20 million. I would assume that's a Series A price round?
yeah.
Okay.
Yeah.
And tell me about the team.
Yeah, our team is myself is the founder, and we have another co-founder, Dr. Hong.
Yeah,
who
can
is
you go to the next slide?
Yeah, 20 years of collaboration history for developing this thrombase SH1, and we are both inventor and author of this technology, and we also have a UCLA team who is an expert of this stroke.
And has Dr. Hong or Dr. Kim, have they ever developed any drugs or for approval?
Yeah, actually myself, I did a major drug development at Northwestern University. And then since then I went back to Korea, and then I did drug development for nanomedicine to treat cancer.
Yeah.
And then I did from discovery stage to the clinical stage.
Right.
So that's what I did.
Yeah.
And so I'm the expert of
So
this
talk
field.
about building of the team beyond the founders. Certainly you're going to use collaborators and consultants, outsource as much as you can. I get it. But who are some of the key people that you need to bring in to run this?
Yeah, since we just founded last year, so we have to build a team, of course. So financial, we need a team, expert in financial, but also most importantly, we need somebody who is expert in drug development. Not development, but just regulation process of FDA.
Yeah.
Because everything is already proven. Efficacy is good, safety is good. We just need to do preclinical development. That's just typical standard study like GLP tox. So it can be done easily. And after then preclinical, after that, clinical just development, IND documentation, and clinical trial design. So this can be done just by expert. We just need to bring them.
I thought this is really interesting. So first of all, great story. So thank you for the great presentation. I guess I had a couple of questions regarding how it's going to be used. So TPA currently it's used, you'd have to rule out the hemorrhagic stroke. So it's only used for ischemic stroke. Do you think this would have indication for both? And it would have to be a hospital setting, right? So I think TPA is only given when you go to the hospital. And then the second question that follows is, I think in the case of TPA, and I'm not a clinician, I think there is a way to actually turn it off. So I think there's an arginine injection that you can do afterwards that basically turns TPA off. Yeah, do you need to do something like that for this, or is it just you inject it and you go?
Let me answer the first question first. This one in the stroke, like 85% is ischemic, and then the remaining 15% is hemorrhagic. But even this hemorrhagic, people think is caused by ischemic stroke just by treating with the TPA. So at the first site, if we treat with ours, it'll be like 100% prevent to convert to hemorrhagic stroke. So, we're going to target both.
Yeah, so I guess my understanding was that you'd have to, and if there's a clinician, they could probably comment better on this. You'd have to rule out that it's not a hemorrhagic stroke, and then you give TPA.
Right.
So what I'm saying is this might be better because you don't need to rule that out. It seems like based on the slide you were showing, which is very interesting.
Your point is absolutely right. When we do clinical trial, we like to include the patient who is not qualified for TPA. So it'll be like the person who arrived after this golden hour, like 4 hours later since the onset, and also the person with a large clot, which cannot be treated well with the TPA, and also the person with the bleeding risk. So of course, hemorrhagic risk will be hemorrhagic ischemic will be excluded.
I guess maybe a kind of just follow-up question. The other issue with TPA is that when you resuspend it, you have to use it. So if it's not used, you have to toss it. And I think every time you use it, it's about $9,000 cost to the hospital. So that could be another really interesting application. I don't know what the stability of there's a serine protease you mentioned, right? If you can formulate in such a way that it stays stable. So you basically, I don't know if it's a suspension or it's a powder that you're working with.
It's a suspension. It's a protein we use bacteria. So it'll be produced by bacterial expression. So it'll be like 100 times cheaper compared to TPA in terms of production cost. And then during the clinical trial, you might think we have to use a TPA as a reference drug, but that's what I'm trying to take it out from our clinical trial design, because we're going to treat a patient who is excluded from TPA. So yeah, we can
Very
take it
interesting.
out. Yeah.
Thank you.
So I'm curious about the pharmacodynamics or pharmacokinetics, excuse me. So how long does it take to realize the therapeutic effect?
Actually, it's going to be acute treatment. So you kind of inject at the site when emergency situation came. So PKD is not really matter in this situation. When you prevent something by injecting earlier, probably PKD is very important matters. But for this acute situation, I think since we're going to just both inject with this drug at the site, like brain or heart attack site, whatever. So it'll be later on, we can discuss more. Yeah.
Maybe just one more suggestion. You should look at Army grants, because I think if you don't get bleeding, that there's going to be a lot of interest in something like that.
Definitely, there is no bleeding, so it will be really wonderful, like ideal drug for stroke as well as heart attack as well as venous thromboembolism. All these affecting 50 million American altogether. Yeah, so many people die every 30 seconds. So we're really trying to push this ASAP. That's why we need.
Is there any competition in development? You mentioned TPA. Is there anything else in development that's competitive with us?
TPA has been the only drug since the last 3 decades, and ours is totally targeting different patients, like a patient excluded from TPA. So even that, I don't think there is a competition. But still, if you treat the same patient, such as a patient who can be treated with the TPA, still, I think people should choose ours, because ours definitely, in terms of efficacy and safety, much superior compared to TPA. So there is no competition. I had to say this, but I know you don't like that word, but there is no competition at all. Yeah. Thank you.
Thanks.